the introduction. So I'm going to share my screen. Can you see it?
Yes, we can see it. We just need to put it on full screen.
Okay.
Perfect. Yeah, I'll just shortly introduce our two speakers. So first we will have Sylvain Glémin, who is from the EcoBio group related to the Rennes University here in France. So he was one of the principal investigators for the DIVERS group, and he is researching evolution of mating systems and the genetic consequence of selfing, the effect on genetic diversity and speciation, and the underlying mathematical models that he then tests on these organisms. And secondly, we will also have a presentation from Mios Käfer, who works at ISEM here in Montpellier in France. He is also a principal investigator for the DIVERS group. He works on plant evolution, sexual systems, and in particular the evolution of diversity, the effects on plant diversity, and the genomics of sex chromosomes. And so, yes, we are very happy to have you here and excited to hear about your presentation. I'm going to mute myself and please, it's your turn.
Okay, so thank you very much for the introduction. So with Jos, we are going to talk with two voices, so about the diverse projects, about plant diversity and the reproductive traits. So I will start to introduce the context where we decided to work on these questions. So as we were interested in the diversity of reproduction in plants, as you may know, there is a huge diversity of flowers and the way plants reproduce, which is the main characteristic of what is called the flowering plants. And this huge diversity of flower morphology is also associated with a huge diversity of species. So there are about 350,000 species of flowering plants, which is around 90% of all land plants. And so it's clearly the most diverse group in terms of species of land plants. And they arise rather recently, actually. It's not completely clear, but the most recent common ancestor of angiosperms, so flowering plants, is around 150 million years ago. It's the same order of magnitude of than mammals, for example, where there are only 6,000 something species. And one of the questions is, why is there such a huge diversity in terms of number of species? Is it just by chance, or is it due to some characteristics, some key innovations, typically like the flowers, but there are also other vegetative characteristics that may explain this huge diversity. But it's also a unique event, so it's a bit difficult to know because there are a lot of synapomorphy of angiosperm which arise. We don't know exactly in which order and at what time before the emergence of the current species. And among this huge diversity in flowers, it can be related to the way species reproduce. And we can be interested in two aspects. One aspect is a diversity of flowers related to pollination, because plants are fixed organisms and they need some vector to put together male and female partners. And typically what we have, it's what we can call the perfect flower, which are mostly hermaphroditic with female and male component, male part on the flowers, and with some attractive organs that can typically attract some pollinators, so typically insect pollinated flowers. But there are also quite a lot of variation, either by having other mode of pollination, like queen pollinated flowers with reduced flower, lack of attractive part, lack of scent, for example. And on the contrary, other kind of flower which are very specialized to attract some specific insect, like these orchids, which can mimic an insect to attract males, and mimicking copulation with this is fake male to pollinate, or the specific red flower with a bell shape, which can be typical to bird pollination. So they use a huge variation on the floral structure linked to pollination. But there is another aspect which is linked to reproductive systems. So not only the way pollen is transported, but also how, from one side, sexes are distributed among within individuals, and what are how the choice of the partner are done during reproduction. So here are some examples. So most plants are hermaphroditic with flower, what I call perfect flower with male and female organs. But some other flowers can have separated sexes. So here, for example, on the same individuals, you have male inflorescence, so only male flowers, but on the same individuals, only female flowers. But you can have also species with separated sexes, like it's quite usual in animals, what we call dioecious species, with male individuals and female individuals. And it can be pollinated by wind. Here it's an example of a tree, or it can be also insect-pollinated flowers. And so this is for the distribution of sexes among and within individuals, but including among hermaphroditic species, or species at least which have the two sexes, so male and female, there is also variation in the way the partner reproduce. So classically, there are what we call outcrossing, so one individual acting as a male and one individual acting as a female. But because a lot of species are hermaphroditic with perfect flowers, so hermaphroditic flowers, they can also self. So in that case, there is only one partner. And there is a range of variation from species which are obligatory outcrossing with some mechanism to avoid selfing, with intermediate mixed mating system, and some highly selfing species, mainly reproducing through selfing, so without additional partners. So what could be the link between this diversity of flower pollination mode and associated reproductive trait and the species diversity? So what is or what was already known, that there are some correlation or association. So typically species which are quite specialized with bilateral symmetry, occurrence of nectar spurs of self-incompatibility system usually increase the diversification rate, so the formation of new species. But the problem is that these traits are also correlated with other traits. So these pollination and reproductive traits, for example, unisexual flowers are correlated with wind pollination. Self-compatibility and selfing are usually correlated with annuality. Dioecious with fleshy fruit. So it's difficult to know what are the possible drivers of species diversity. And another point is that there are actually many exceptions. So it's difficult to draw a general rule about the link between diversity of reproductive traits in general and species diversity. So the question we asked in this project was to try to understand how do the different traits can shape plant diversity, trying to take them together. And the plan of the project was first to try to predict what should be the effect of the different traits on species diversity. So to try to have a theoretical framework or a conceptual framework to link reproductive plant trait diversity and species diversity. Also to try to summarize what have been done and what is observed in the data. And finally, because we see that there is some possible correlation and the diversity of structure participating to reproduction, to analyze the co-occurrence of floral and other traits and the link with reproductive systems. And to do so, what we've done is to gather this diverse group. So there are 15 to 17 people, because that's a nice thing with the Cesab. It's quite open. We can start with a group and invite other colleagues. Some have left also for different reasons, because one colleague moved out of academia, for example. So it's very open and flexible. And what we try to gather, so it's a diversity of persons, so from 10 countries approximately, and with different backgrounds. So people coming from specialists in evolutionary botany, evolution of plant reproductive systems, but more on empirical part, ecology, evolution, and also theoreticians and statisticians. So that was the richness of the group. And as Nicolas mentioned, the fact that we could gather several times a whole week at the Cesab in Montpellier was really excellent to have a real group functioning with a really nice interaction and fruitful interactions. And among the group also, Andrew Amstetter was a postdoc associated with the group, and he did a really excellent job to maintain this cohesion of the group. So I'm going to present the first part, which is linked to this idea of trying to get a conceptual and theoretical framework to go from trait diversity, and especially reproductive trait diversity, to species diversity. So what we decided to do is to think with this kind of scheme. So actually, species diversity comes from the diversification process, and diversification, basically, it's simply a speciation minus extinction. And the question is how plant trait can be related to these big processes, so speciation and extinction. So here it's represented here, speciation, and instead of extinction, we call it the reverse, so persistence probability, just to have the sign in the same direction. So speciation, higher speciation leads to higher diversification, and higher persistence leads to higher diversification, so more species also. And what we've tried to do, because many evolutionary and ecological processes can affect speciation and extinction, we try to define a main group of processes, and we got to this classification with effect of the speciation being affected by two main processes, which is divergence in terms of trait, phenotype, and etc., that allows coexistence of different groups of individuals, but also reproductive isolation to, at some point, remove all possible gene flow between these two groups of diverging individuals. And for persistence, which is the contrary of extinction, we identify two main mechanisms. One, which is more a short-term mechanism linked more to ecology, which we call demographic buffering, the fact to be resistant to perturbation, to have, for example, high population size or high growth rate, and evolutionary buffering, which is a way to persist on the long term and to cope with variation in the environment. So basically, we can call this also the adaptive potential of the species that can persist through long evolutionary time. And then what we try to do is to look at different plant traits and ecological contexts to identify how this trait can affect divergent reproductive isolation and so on. So to do that, we did a literature review mainly on theoretical models and conceptual models, so not on data. So we clearly avoid for the first part to look at the data, just to have what could be the prediction, to have a hypothesis-driven approach. And we identify 13 traits that could be important linked to and linked to reproduction. So here I give a first example, which is one of my favorite examples, which is selfing. And here things get quite complex because selfing is mainly the most emblematic example with many effects on both the genetic functioning of a population, but also the ecology and demography. So here the point is not to read all the arrows and the effects, but you can see that selfing through many mechanisms can affect reproductive isolation and divergence in many ways, and evolutionary buffering and demographic buffering in many ways. And at the end, we don't get a clear picture because for the speciation part, it's difficult to predict whether selfing will increase or decrease speciation because there are antagonistic effects for both reproductive isolation and divergence. And for persistence, so the contrary of extinction, it's the same. On the short term, selfing produces reproductive assurance, so which can be a good way to allow the population to persist, for example, if there is no pollinator in the environment. But on the long term, this reduces the adaptive potential of the population, so can lead to, on the contrary, higher extinction. So it's difficult to predict what should be the effect of selfing on diversification. And actually, when we did that for all the other traits we chose, it was roughly the same pattern. So basically, you see you have plus and minus in many directions, so it's difficult to have a theoretical prediction of what should be the effect of a trait on diversification. So globally, most traits, if not all, have contrasting effect or opposing effect on diversification. And in addition, some traits are correlated. So this also complexifies the prediction. And the net effect should depend on the balance of this interaction. And one way to have an idea what predominates is to look at the data. It's what can the data tell us about this interaction, whether some of them are more prevalent than the others. So it's what we've done in the second step. So this was driven mainly by Andrew. And the idea is to use approaches using phylogeny and diversification methods testing the effect of a trait on diversification and to review all this literature to try to have a picture of what really happened in nature. So in this method, which are called state-dependent speciation and extinction models, basically the idea is having a phylogeny and a mapping trait on the phylogeny to test whether one trait, so for example here the red trait, increase or decrease diversification compared to the black trait. And there are quite a lot of models for that, for binary trait, quantitative trait, geography, and so on. So what we've done is to review the literature of paper using all these kind of models. So here you have the histogram of the increasing number of studies using these models through time. So since the origin of this, the first methodological development. And from that, we retrieve what were the traits tested by the authors and what were the effect of these traits on diversification. So the main picture is that basically there is no clear pattern. Here, it's this histogram show on the x-axis the big category of traits which has been tested. So reproduction is one of the main traits tested, but biogeography, which is not really a trait, but a characteristic of the species, an ecological characteristic, so biogeography, vegetative, and so on. And what you see here is that roughly the whole combination, you see that there is no category affecting clearly diversification. Half of the study shows there is an effect of the traits on diversification, and half of the study shows there is no effect. So there is no single trait or single category having a consistent effect on diversification. And when you look at more clear in details, you see that clearly depend on the evolutionary context, the ecological context. But another important point of these studies was to look at the more the statistical part and the properties of the data sets that have been used to look at in these studies. And here it's some example of the dataset characteristic, like the number of tips in the phylogeny. If you have a big phylogeny, you have more power to detect an effect. But you have also some things quite strange, like this sampling fraction, whether you have many missing data, so you are in this part of the tree, it's easier to find a positive effect of trait on diversification. And this can be due actually to false positive because you don't sample very well the phylogeny. And there are some strong effect of the characteristic of the data set on the results. And what is interesting is that only 20 studies over all what we gather really meet the quality guidelines proposed by the authors of the method. And finally, if we combine the biological results and these more statistical results, what we found using an approach to predict what should be the effect of whether we can obtain a positive effect depending on all the information about the biology and the data sets. The first characteristics are the data set characteristic. So the data set characteristic are a better prediction of an effect on diversification than the biological characteristics. So this means that it's really important to have good quality data sets because before trying to draw a general conclusion. So this was the two first parts, and these two first parts lead us to the conclusion that it's important to take into account the context and also to see that not a single trait have a consistent effect. So also to have an approach trying to gather multiple traits and to have a multivariate view of a reproductive system. And Jos will present these parts. So I'm unsharing the slide and let Jos continue.
Yes. So we saw lots of things on how to study diversity, plant diversity, either with models or theory or with data. Nothing really clear comes out. So, well, we had to look ourselves more clearly into data. So, so we asked, it's already clear from what we saw, like, what actually are we doing when we're studying the effect of traits on speciation? And actually, very often, what we are doing when we're looking at plants, at plant traits, is looking like this. We take the plant apart, decompose it in certain traits, and then, well, we will try to see what affect the diversity. But like plants, you know, they are like this. They are not just a simple collection of traits. They are together in a way that they occur together in a way that's not always easy to quantify. They occur in a natural environment. So to try to tackle this in more to better tackle this, I'm sorry, I can't. Yeah, okay. We went to look, mostly from evolution. We try to look what has been done in plant ecology, which is looking at lots of traits. That's what ecologists do, right? So what has been done in plant ecology? Well, people in plant ecology have defined ecological strategies. So here's a very famous one, the plant ecological strategy, which has been proposed by Grime a long time ago, that plants actually, the species composition of at some point will be determined by competition, the stress and disturbance that the plant community undergoes at some point. And this has been later linked to certain plant traits by Westovi, for instance, specific leaf area for competition, plant height also, and the seed mass. There's a link to these ecological variables. So this has been a very successful approach, and now this is a paper from a few years ago, not from us. These traits, so leaf, height, seed traits, have been collected for a large number of species. So this paper was published a few years ago. It had data on about 46,000 species, and actually shows the same thing. There's a height axis and there is a leaf axis. And what is quite striking that this actually explains quite a lot of the variation in plant traits. So 74% of the variation has been captured in these two PCA axes. So we wonder then, well, where do reproductive traits fit in here? And people have identified or termed reproductive strategies also using these kind of data. And this is a very influential one by Salguero Gomez, published a few years ago. And you see here what he calls reproductive strategies. But if you look at what reproduction is here, then it's mainly like how many seeds do you produce, how many offspring do you produce? And that's also, well, of course, clear if you look at the ecological databases like the one that the previous paper that the PCA was had been using. So this is data from TRI. They have 12 million of traits for lots of plant species. But you see that the largest part of these are actually leaves or whole plants. And there is some part about reproductive organs, but the reproductive organs, that's mostly seeds. So we asked how we can fit flowers in these frameworks, because we know flowers are linked to evolutionary diversity, species diversity. They have important ecological roles. I think nobody will deny that. So you can't have seeds if you don't have pollination in the flower first. There is pollinator competition, but among species, it also leads to species coexistence. And also is very important is then pollination, and the way you're pollinated also influences the outcrossing. It influences genetic diversity and the capacity of adaptation. So can we integrate these flowers and what happens actually before the seed is produced into these reproductive strategies? So now just take a step back to go to diversity. We've shown some pictures of flower diversity, and it's like, well, much like comparing, as we say in France, comparing apples and pears, or apples and oranges, or bananas and oranges, or well, we want to compare those. Well, actually, flowers are easy. Fruits are easy. Pears and apples, they are easy to compare. For flowers, it's much more difficult because there's so much diversity, right? So here you see just two kind of standard flowers. They have petals, stamens, etc. Although botanists would not say that the first one has petals, but okay, let's leave that aside. They have petals, stamens, stigmas, etc. So they're kind of comparable. But then you go to more subtle, more spectacular aspects, not subtle, spectacular aspects of diversity. We have these orchids that have very peculiar shapes of the petals. You also have these these sedges down here that are wind pollinated, but they actually don't have petals. So how do you compare those? And then another level of complexity is added when you look at how these flowers are organized, because here the two things that people would recognizes flower actually are inflorescences. There are still a bunch of flowers together that actually determine their attractivity and how they're pollinated. So there is this incredible diversity that we want to describe. So for this, we sat around a lot with the people involved in this project to try to come up with a data set that we could use to test how actually these co-vary. So we have been working, our goal was to work at the angiosperm level. And so we have 300, we gathered a data set of 360 species, and it doesn't look like a lot of species because like the angiosperms is 350,000 species, but we were kind of careful to select species from the most important families. So we have all families for which there are more than 100 species. So that is 0.1% of angiosperm species diversity, but 41% of the families and 95% of the plant orders. Then we defined the plant traits together as well. While discussing in Montpellier, define the plant traits we want to gather information on, like classical life history traits, but also the mating system, self-incompatibility, and then the more specific flower traits that we wanted to get. So we defined those, and then we started to put them into our database, which was provided by our colleague Hervé Soquet, the Proteus database, where you could actually collectively work and check what others are doing, which allowed also, which was an important part of the project as well, because remember, we ran this project from 2019 to 2022, which was during lockdown as well. So people from different labs in different countries could also participate in entering these data. And well, it was a huge effort, even if the data set doesn't look that spectacular, 360 species, sorry, because you have to get all this information from floras, from specialized literature, and they're sometimes very difficult to find. So in the end, we had like about 10,000 entries, they were tracked and referenced. And then, and this is here also where Andrew Helmstetter did a lot of work on trying to analyze these wealth of data. So I'm not going to go into detail. I just want to mention some of the challenges of biodiversity results. If you want to actually reduce the diversity to allow comparison, but not too much. So we reduce the categories, we split the traits, etc. And then to analyze this, so we have different kinds of data, numeric and qualitative. So you need to appoint some special methods to actually take care of all this correctly. So what we got then in the end, or what Andrew got in the end, was this PCOA, the principal coordinate analysis of the species. So the first thing that struck us actually was that in the first two axes of variation, we only have 30% of the variation in all the data set, which is very impressive if you compare it to what's present in the vegetative traits. So kind of enhances this idea that there is a lot more variation in flower traits than there is actually in vegetative traits, which is not really surprising, but it's important to have it quantified at this level. And then we defined two major axes. We found two major axes in this trait space. One is like from the diagonal from left at the bottom to right at the top. It's a kind of traditional lifespan and size and woodiness axis, which is like related to vegetative traits, but almost perpendicular to this is a pollination or floral attraction and axis, which comes up in these first two dimensions. So now we wanted to look where our different reproductive systems were. So we have dioici in kind of red. So the unisexual individuals, the yellow with the unisexual flowers in the same individual, that's monoici, and then the outcrossing and the selfing species with the group also a mixed mating system in between. And we see that they can actually quite well separate in these axes. especially for the outcrossing part. There is a clear separation from selfing at the top right to outcrossing at the left bottom. But this outcrossing part is separated into species with hermaphroditic, so bisexual flowers that outcross and that are showy, and the dioecious species, which are much less showy and have mostly wind-pollinated species. So the selfing is concentrated in the part of the trait space where there's only the short-lived species. And the monoecious species actually are kind of in between. So this gives us some idea about the evolutionary and ecological forces that are at play here. Of course, this is a classical result in evolution that outcrossing is associated to size. We have these large, long-lived plants that suffer more from inbreeding than small, short-lived plants, so they should really outcross, while the small ones can also self. And then this is also where the ecology then kicks in the outcrossing, because there are actually two very different ways to achieve outcrossing. So either you develop highly attractive and rewarding bisexual flowers and to rely on insects or mainly insects or other animals that transfer the pollen and that lead to outcrossing, or you have smaller flowers that are low rewarding, and then you could evolve separate sexes. And it doesn't really matter if it's biotic or abiotic pollination. It's really the flower itself that determines if you go this route. So we know that outcrossing evolves to avoid inbreeding, but how it evolves it depends on ecology. So this gives insight in further theoretical studies that some people are actually doing at the moment, and also it highlights the distinction between the bisexual. No, the dioecious. I made an error here. Sorry, the dioecious and the monoecious species is actually quite poorly understood. The bisexual as well, because the monoecious species are in between. So why do some species become monoecious? That's still an area of investigation. So there is also more diversity here in this trait space, and we can visualize that if we look with different methods like nonlinear projection or different clustering methods. So if we do this nonlinear projection, we see three groups, species with unisexual flowers, bisexual herbs, and bisexual trees. But if you go in with clustering, to look more into these groups, you also see that in the bisexual herbs, there is floral symmetry. It's like the actinomorphic and the zygomorphic flowers, which clearly sets them out. This is indicated by these two colors. And among the trees, it's the way you disperse seeds. And so it also points to the fact that more patterns are to be found in when you go to smaller scale analysis. And finally, then there are the exceptions. And I think this is also very important to realize when you're doing this biodiversity research. So this is a slide that has two goals. One is to highlight the importance of Hervé Soquet, who is the botanist here to this project, who actually made this database and was very instructive for this project, but also well, the botanists might recognize it, maybe non-botanists as well. This is a Rafflesia species. I just told you that dioecious species, so with separate sexes, usually have very small flowers. They invest little in their flowers. Well, actually, this Rafflesia species is the species which has the single largest flower on Earth. It's dioecious, so it actually doesn't fit our expectations, our predictions. But also, it's like we have to be humble as botanists of studying diversity, because this species is very difficult to compare. It has a flower, so we can compare its flower to the rest of the flowers, but actually it's a parasitic plant that doesn't have leaves. So how to compare this with other species on this very qualitatively different part of biodiversity, that still remains an area of investigation. So to wrap up, I So we're working with this towards a description of functional flower diversity, which is we still have quite a way to go. And the goal that this would achieve is to actually put more together these fields of ecology and evolution that we feel as, at least coming from evolution, have been quite distinct. Evolution has focused on adaptation, speciation, extinction, ecology much more on competition, the interaction with the environment, and species coexistence. And like a message we want to convey here is that if you want to combine these, and this needs to be done, looking at flowers is a very nice way to do this, because actually they're at the intersection of this. They interact with insects in pollination. They also affect outcrossing, which will affect the evolutionary potential of the species. So this is especially important in this current context of biodiversity crisis. We actually have an decline in insects. So species are suffering from not being pollinated, or maybe changing their pollination syndromes because insects are declining. And in the climate change is going so quickly now that species have to change, they have to adapt. They can't just simply sit there in their environment and do if nothing happens. They have to outcross and to adapt to the climate that's changing. And this is, well, achieved through pollination and through flowers. Well, needless to say that much work needs to be done to achieve this. So as Nicolas mentioned in the beginning, so we followed us. We have been trying to extend these projects to others. So either at the FRB, so the Foundation of Research on Biodiversity, or a French National Agency project. And also we're preparing for a European doctoral network now, also in which the Cesab will be involved. So there's no conclusion about, it's not the end of this research. I think it's an important aspect of the Cesab groups, that you can actually put people together and try to come up with a framework that will allow more research to be conducted and to better understand these biodiversity patterns. So with this, I'll just like to thank all the people that were involved, to mention them here with all their names, especially Andrew Helmstetter, who was a postdoc in this project. Also wasn't that easy, because a month after he was hired, it was the lockdown. So he did a good job in keeping everybody in contact as well, and doing this research. And of course, I'd like to thank the staff at Cesab, so Nicolas Moquet, Maud Calmet, and Nicolas Casajou, without whom this would not have been possible. And thank you for your excellent support. So this is it, to be continued, and we'll be happy to have your questions on any of this.
Excellent. Thank you very much, Jost and Sylvain. That was super interesting. And so, yeah, now we will take some time for questions. So those of you in the audience who have questions, please raise your hands using the hand raising tool that you can see at the bottom of the screen. And I will distribute the questions based on who raised their hands first. And if you're unable to talk, then you're welcome to, or invited to, write your questions down in the chats. We'll pay attention to that as well. So yeah, go for it.
Well, I might break the ice. I mean, that's my job. Thank you very much for this very interesting talk, and I'm pretty far from that field. But from the first part of your presentation, when Sylvain, you showed that you found as many studies finding an effect than not finding an effect, and I was wondering if you had some ways of weighting the redundancies between studies, because you might have some studies that actually tested the same.
Yeah, so thank you for the question. Actually, what I've just presented the very general view, but what we've done also, it's for the different traits to look at the studies that tested the same or similar traits. Actually, it's not that much, so it's also that's an important point. It's good for this analysis to repeat the same kind of analysis in different groups to get some robust results. And actually, most of the case, it was not consistent, except two traits. So outcrossing versus selfing, but it's not a lot of studies, but all studies point to a higher diversification in outcrossing species, which is partly predicted by the theory when you better understand the weight between the plus and minus. And another, which is not which is a vegetative trait, is C4, C3 photosynthesis, with C4 photosynthesis globally and quite consistently increasing diversification. But for example, dioichi, there's quite a lot of studies where, or at least it's one big paper when they repeated the analysis several times. And here it's completely, basically it's half of the result is in directions. So it seems that some traits could have a consistent effect, even if theoretically it's difficult to predict what should be the direction. But others have no consistent effect.
Just a single precision. So you've been doing a kind of a map of what is expected based on theory. And so did you actually found some areas, I mean, some predictions that haven't been tested and that should be? Like to release, you know, to give some some food for scientists, you know, to do more.
This is a good point. Actually, yes, there are some traits which are not properly tested. Actually, we also did this and use this approach because in this kind of analysis of diversification, some are clearly based on hypotheses to test, but many are just exploring the diversity and the trait that could. And we wanted to try to have a conceptual framework. Otherwise, it's easy to test many things and to give some explanation a posteriori. So we wanted to have the reverse approach. And it's an opportunity also to mention a side project issued from the Cesab group, which is quite difficult, so it's still ongoing. It was the idea in parallel to have a database of reproductive traits. We have started, especially Sylvain Billiard, started to develop a database of model prediction. So to have the kind of the parallel between what the model predicts and whether there is consistency in the prediction of the different models. And some predictions are very clear and supported by many things, and others are also not clear. And also some model have never been done. So it's also a way to point to what could be interesting to develop. But it's quite a difficult task to conceptualize such a database, but it was also linked to this DIVERS project.
Works for years. Okay, thank you.
Are there any questions? Otherwise, I have a few. Well, I had questions or remarks, things that I found that appealed to me in your talk, and I really like the part, Jos, when you were talking about functional traits in flowers and how when we're measuring traits on flowers, but I guess it's true also for like any other organisms, is that basically you're taking them apart and looking them at and as if they were deconstructed. And I was wondering, I don't know, this is also kind of the purpose of measuring functional traits in the first place. And so I guess my question is, how do you go about this? And are you like looking at interactions of traits, or like, I guess, yeah.
Well, this was also a kind of I don't know if many of you saw the Cesabinar that Cyril Viol did a month ago, saying, well, okay, Cyril Viol is really the specialist on functional traits, saying, well, okay, there's lots of functional traits we can measure, we can compare, but there's also a large part of diversity that's going unnoticed, right? In these kind of analysis. So I think what he mentioned was a stink bird, which is so unusual that you can, like, it doesn't really compare to any other organism. So it's still too early to give a definite response to answer to this question, because lots of the work we have been doing, and well, we're not the only ones. So other people as well is trying to actually figure out how to code, how to compare, how to measure, how to and how to study all these traits. So keeping the diversity. So keeping the diversity while still having something to study is a well, maybe even a philosophical question in
exactly.
how you study biology, right? You can press everything together in a few traits that you can measure, but then you don't understand the diversity anymore. So so I don't have a definite answer to your question, and I think this is also a very important part of what's happening in this Cesab universe and the Cesab ecosystem. It allows us to discuss with people that also have similar research questions, and we can advance together with. So Cyril was presented to us when we had our first meeting at Cesab, and he will also take part in projects that we will develop furthermore. So there's still a lot of things to do.
For sure. And just a very small question, maybe I missed it, but did you also measure traits that pertain to the, I don't know, physiological aspects of flowers, like chemistry, or maybe, I don't know, compounds of pollination?
No, we didn't, because that was also one of the goals of the project, is really to make a database that representative to angiosperms. And then, as you probably know, lots of the angiosperm diversity is found in countries where there are a lot of researchers. So getting actually a lot of data that you can compare between a lot of species, we also needed to gather data on on species that only occur in some tropical places where people never go. But what's available always is the description of the species with some botanical information as well. So that was our goal, to actually have something comparable. And so we started also because we were interested in outcrossing. So we started with species that had some outcrossing data available. So we couldn't add more, but yeah, indeed there would be room for people to go to all these nice places, but that's not funded in the Cesab project, but to go to all these nice places and measure traits, and actually to see how physiological traits might be related to other morphological traits that would be easier to measure.
Right. Thank you.
So I hope it didn't scare people too much, but
well, then I guess that's it then. Or maybe Xavier.
Yeah,
Xavier would like to ask a question.
It's just an idea. So there's lots of interest from trying to put links between the micro and macro evolution. It's something that we are hearing all the time, and I think there are many different approaches to trying to do that. And here, I think it seems that you are focusing on macro evolutionary patterns, trying to, but also the what Sylvain has presented on the effect on divergence, etc., are also linked to micro evolutionary patterns. And so I was wondering whether it would not be interesting in parallel to gathering all this data, but also to trying to set up some population genomics data sets, or to which could help maybe to find other ways to test the effect of these different different plant traits. And so I was just wondering whether this is something you have in mind, or you think it's not your business, or I don't know.
Maybe I can actually, it's what we're going to try to do in this new ANR project. It's exactly so basically was to try to link this macroevolutionary and multi-trait pattern with data both on short-term ecological patterns. So, for example, with data on species abundance, community surveys, things like that, and also genomic characteristics. So for the moment, rather basic characteristics like measure of genetic diversity, of general mutation load, of recombination, things like that. But this is the idea, to try to inform macroevolutionary analysis with micro evolutionary processes like genetic diversity and so on, and also ecological process. I don't know if we will manage to do it, but it's exactly the plan of the ANR project we have just started.
Yeah, I think it's really the way to go to put together these two approaches. Yeah. Thanks.
Thank you, Xavier. Any other questions or remarks? Would you like to add anything, Jos or Sylvain, especially related to the functioning of a Cesab group? I mean, you talked about it already, both of you, in your slides, and depicted how it was a particular situation being a Cesab group during the COVID lockdown and everything. I don't know, is there anything else any tips or any pitfalls that you would like to share with us and maybe other people that are part of Cesab groups that might be in the audience, that, so that they wouldn't do the same mistakes, or that they can benefit from your experience?
So maybe the first thing is just to say that Cesab is really a great place to do science, this kind of science.
That's
and it's
exactly
really
what we want to hear. Thank you.
It's really a unique place to do that, and I think with the group it functioned very well. So yeah, and it's really because we have a kind of 1 week of free thinking, and with all well organized around this, and yeah, it was really I guess all the members of the group also enjoyed a lot. We hope so. But it's really a unique place to do this kind of research. And yes, you, Maud, Nicolas, and so on are really great to support us. So it was really a really great experience.
Yeah, it's a great place to have new ideas, but then actually to have your research agenda filled for the next few years, because all these you have to test them with data and with other hypotheses. Also, Xavier suggested, so like there's lots of links to make between what you observe in diversity patterns, what you observe in ecology, and also in microevolution. So. I think thanks to Cesab, we will be busy for the next, I don't know, N years working on this kind of projects. Also, maybe just to add, it was also nice to be two, I think, to do this, to manage a group of 15 scientists. It's not always easy, so we were two to do this, and also actually three with Andrew, also the postdoc who did also a lot of help in actually getting all the people together. So scientists are not the most the most punctual people, and so it's good to have some people to actually guide this into the right direction. But like, especially when you're together at Cesab discussing all this, ideas are going really fast. So some guidance is sometimes needed.
Well, it's really nice to hear that you had a good time at Cesab, and like Nicolas said at the beginning of the Cesabinar, I guess it's a shared appreciation. Do you want to add anything, Nico, or does anybody have a question before we finish?
Just want to thank everyone for having attended today. We were at 70 at the peak of your talk, which was a very good number, and so I'm glad that people were interested, and I want to thank you again very much for giving us your time back, and I would like to thank you, Camille, for organizing the Cesabinars and making things possible. Thank you.
And thanks everybody for attending. And so this video I will stop the recording, and this will be online on the YouTube channel of the FRB for those of you who want to watch it again or share it to your colleagues. Thank you very much.
Thank you.
See you in a month. And thank you, Jos and Sylvain.
Bye.